Welcome to my lecture on genome and genomics. My name is Carsten Daub. I'm Associate Professor at the Karolinska Institute in Stockholm. You can find some more information about my lab at daublab.org. The day of this recording is January 26, 2021, and the recording was done for the course KB7016, which was given in 2021. Today I will talk briefly about some of the topics listed here, which is genome sequence, genome annotation, gene models, something on data on the genome, and something on RNA sequencing. And the basic learning outcomes is that you would be able to describe basic principles of the genome sequence and genome annotation according to the topics on the left here. I will start with some basics on the genome sequence. So to make sure we are on the same page. So this example here is on the right side are the chromosomes of the human genome. There are 46 chromosomes. And so if you would determine the DNA sequence of the human genome, you would basically determine the DNA sequence of these 46 chromosomes individually. What is important to keep in mind is that if you would sequence the DNA sequence of the human genome, you would sequence it from one specific individual, because you would sample tissue from or DNA from one specific individual, and you would sample that at one specific point in time. So whenever you think of a genome, then you have to be aware of that. This is the genome of one specific individual of a certain species. And that is at a certain point in time. And that is important, of course, because genomes change over time, so to speak. So you would have somatic mutations, which are the mutations that occur in the genomes of some of the cells. And that depends very much also on the tissue you're looking at. Some tissues are much less prone to somatic mutations. So for example, bone marrow would be maybe less prone, whereas other tissues that are much more exposed to the environment, like skin or other things, would change much more over time, because they are, for example, exposed to UV radiation and so on and so forth. Also, mostly when we talk about genome sequence, we are referring to the nuclear genomes of cells. And the nuclear genomes are the genomes that are found in the nucleus of a eukaryotic cell. And it has to be, you have to also understand that there are other genomes, most often genomes of cell organelles. So for example, if we think of eukaryotes, of humans, then we would say that the mitochondria are organelles in the cells, and they have their own genomes. The ribosomes also have their own genomes. And if you go to plants, for example, there's the chloroplasts, which also have their own genomes. Interestingly, some of the, for example, ribosomal genomes, for example, some of the ribosomal genes, they are actually also present to some extent in the nuclear genome. So there have been some exchanges over time, but in principle, these are different genomes. So when we talk about the genome sequence, then we talk about the sequence of the 4 letters AGCT for all these chromosomes, and we would not consider the meaning of these letters at the same time. So the genome sequence is simply the sequence of the 4 letters in the respective chromosomes. So when we talk about which parts of these sequences are having a certain function or whatever, then so the functional elements, then we would talk about the genome annotation. So the genome sequence in the first as such as kind of can be seen independent of the meaning of the genome, independent of the annotation. How many genome sequences have we determined by now? So I went to the National Center for Biotechnology Information, NCBI, and I was looking at how many genomes can I look at or could I download. And you see on the screen we have a total of around close to 60,000 genomes that are available. And it's 15,000 from eukaryotes. It is prokaryotes, 30,000, and viruses and plasmids and organelles. And this is just the beginning of the list, and you could click further here. You see the sizes of the genomes and some more information about on how many chromosomes. And if you would go into one of those, and I just did this for one virus here, you would come down to a display which looks like this, which is the so-called FASTA format, which you have seen, I'm sure. So you have a descriptor here, it's some virus, and you would see the sequence here, and it's cut off, so it would be much, much longer. Yeah, so around 60,000 genomes are sequenced as of today. Looking a little bit more specifically into the human genome, you would see that the information about the human genome is organized by chromosome. And you see the 22 chromosomes here, and then you see X and Y. And then you see two more rows. One is the mitochondrial genome, which is a kind of organized on its own. And you have a section here of contigs or sequences which are not placed into any chromosome because we don't know basically where they should be placed into which chromosome. You also see that you have only one chromosome, one file per chromosome. So there's no, even though we have two chromosomes, there's only the sequence of one. And so when we here think of the human genome, we should ask ourselves, this is the so-called reference genome. So we have to ask ourselves, who is that individual actually? And that is something maybe you can take as a homework if you don't know already. You can ask yourself that specific genome, which we call the human reference genome, because as I said, it kind of should be obtained from one individual at what point in time. So what is that human reference genome? You also see that the chromosomes are sorted by size here, but that's like a minor detail, more or less by size. I wanted to bring to your attention this fact about the genome size and the number of proteins that are encoded in the genome. That's interesting. You see in this graph, it's a logarithmic plots, log, the genome size on this axis and the number of protein coding genes. And you see that there's a section here where you have a pretty nice linear relationship on the log scale with the genome size and protein coding genes. But then you see that for eukaryotes, which are up here, that it's actually not linear anymore, but actually the number of protein coding genes is very small. Maybe you can think a little bit about what that means. Why are these genomes so large, yet the number of protein coding genes are not kind of increasing with the same size of the genome, with the size of the genome? Please think about that. Yeah, the most recent version of the human genome is version 38. It's also something one can know. Okay, how would we determine the sequence of a genome in the first place? So I will be very schematic here. So you would extract the DNA from the nucleus of a certain sample. What we then do in this approach, which is the short read approach, is that we would fragment the DNA by sonication or with chemicals. And then there's a step where we would amplify these small pieces. And this amplification can be done in several ways. A very common way is an in vitro amplification, which basically puts together these two steps here. But that's a technicality which is not important. So after the fragmentation, we have fragments of a certain size, and we can determine how large these fragment sizes should be. And then we can basically sequence these fragments. And since this is a short read technology, we would usually sequence around 150 base pairs from either end, and this is displayed here. So this is the one of the fragments which are shown here. So you would sequence around 150 base pairs on this end and 150 base pairs on this end. And this old slide, it says 35, 35, but nowadays it's a bit longer. And you see that you would not necessarily sequence the whole fragment, but two ends of the fragment. So it's a paired end sequencing. And maybe the Illumina technology is the most frequently used these days. So you could also then have, you could generate fragments of varying sizes. So some fragments could be, for example, several kilobases long. You sequence both ends. And you would also, you could generate in a different sample from the same individual fragments that are much shorter, and then you can perform a paired end sequencing of those. And you know the length of the fragment, and then you know both ends. So that will be helpful in the end when you compare that to a reference. So here you see an alignment of these sequences to the reference genome. But this is a very complex process, and I will not go into how the alignment and the assembly work, but you can get an idea. If you can puzzle it together like this, you would be able to build together a genome sequence from here if you have a good coverage over the whole genome. Also, if you don't have a reference genome available, you can make a de novo assembly, and the puzzle is just of a different kind. Yes, there's also ways to perform a long read sequencing. So basically, you could have fragments which are not like a kilobase or 2 kilobase long, where you sequence the ends, but we can perform sequencing with the Oxford Nanopore or PacBio, where you can sequence stretches of several kilobases, which could be maybe 10 or 20 kilobases long at times. So then you would have very, very long sequences, and that would basically, it's much less puzzling and would be much more clear how that sequence looks like. And you can combine both of those, of course. So you would maybe perform some long read sequences where you would have a certain number of long reads for the genome, and then you can perform more details by aligning these short reads to the long reads, and you can maybe concatenate long reads that way. Yeah, these are the basics of the de novo genome sequencing with short reads. I'm continuing with the genome annotation. Briefly, you could say that genome annotation is what is encoded in the genome. What does a certain stretch of DNA mean? What is the function of certain parts of the DNA? That is kind of the description of the genome, the annotation of the genome. One of the very basic things that we want to recognize on the genome are the genes. So the genes are some of the functional elements. I think we will not really discuss the definition of a gene, but some definition. You can think of what is the definition of a gene. One way of defining a gene is that it's a functional element that's located on the genome that is also inherited. So there's definitions of genes which are not really related to the genome itself. This is before the actual structure of the genome was known, and so people defined genes as some abstract unit which is somehow encoded somewhere and it's kind of transmitted through the generations. So there's older definitions, but the new definition it would be a functional unit on the genome that is inherited through the generations. So different organizations are defining genes, and some of the examples are the NCBI RefSeq database, reference sequences database, and there's also others like the GenCode Consortium. I put references here, and the University of California, Santa Cruz, UCSC, Genome Browser group, and they are also defining UCSC genes. It is important for several, like for all of the, whatever gene model you are using, it is important that you understand how the genes were defined by these respective groups. For example, for reference sequences, RefSeq genes are basically genes that have been described in publications. So for each RefSeq gene, you get a reference to one or several papers that have defined that specific gene. It's often papers that are specific on a certain gene, where groups have sequenced that gene. So they have very clear ideas about the gene start, the gene end, the gene function, the structure of the exons and corresponding introns, and so on and so forth. So it's kind of a publication-based definition. Other definitions, for example, by Ensemble, by the European Bioinformatics Institute in the UK, they define genes in a different way. They have complex algorithms, which are like pipelines, which are collecting a lot of primary data. And then they use these primary data and combine them in clever ways to come up with genes and also transcripts. And they come up with a much more fine-grained way of having genes with certain levels of confidence. So for example, genes can be like they can have a certain status that's kind of, we are not so sure about this one, or we're very sure about this one. Genes can have a certain coding, a protein coding potential, and so on and so forth. So the approach of RefSeq is to have a relatively small set of genes with high confidence. The approach of Ensemble is to get as many genes as possible, but for each gene to give a certain level of confidence, so that the RefSeq genes would be part of the Ensemble genes, but Ensemble would have additional genes which are not in the RefSeq model. So depending on your question, you would have to choose which model you use. Or you could use the Ensemble genes and you only use the reliable part of the Ensemble genes, which would more or less be equal to or equivalent to the RefSeq genes. So point here is there's different models, different ways to define genes. There's different models, and please understand and read up on the respective model you plan to use. You can visualize these genes on the genome. And one, maybe the most popular way of doing that is to use a so-called genome browser. I give examples here of two genome browsers. One is the University of California, Santa Cruz, UCSC genome browser on the top here. And the other one is a genome browser made by Riken in Japan, which is called Zenbu. So you have basically a one-dimensional representation of the genome. So here we are on chromosome 11, and here you have the coordinates. You see with the scale bar that this is a 1 kilobase window, so the whole window here might be 2 kilobases long. It's centered on a specific gene, and this is the hemoglobin gene, the human hemoglobin gene, just as one example. You see that there is one track here, which is the RefSeq gene, and you see that there is one gene shown here. And then you have another track, these are called tracks. You have another one, which is the GenCode model. And you see that GenCode is giving 3 different models. And you see, first of all, that you have exons here, the darker areas, boxes here, and you have introns, which are these lighter areas in between. And the arrows indicate the direction. So the direction is from right to left here, which is on the negative strand. You also see that the boxes are smaller up here, which are the untranslated regions. So this is a protein coding genes. So the right part here is untranslated region, and this is the 3' untranslated region down here. You also see that there's genes nearby here, and you see that there's 3 different transcripts here, and they have slight differences in the model. So you see that you have a slightly shorter 3' UTR here and here, you have a longer one here. And this transcript is entirely composed of 2 exons. So it's skipping the 3rd exon. And yeah, so you see differences here. When you go to the Zenbu browser, it's basically the same information with a different layout. You have it's the same gene, it's the HBB gene. You have chromosome 11 here, and you see the whole. This is another gene model, N-trait gene, that goes from here to here. It's on the minor strand, of course, the same gene, same information. You have the GenCode gene as well, which is slightly longer here upstream. Here you have transcripts. So you have GenCode has one gene model here for the whole locus, but it has transcript models for 4 different transcripts here, I see. And you see there's a slight difference. For example, the second one is missing the first exon, and so on and so forth. And you also have something that I will maybe bring up in the next slide, which is you not only have models here, which are generated from primary data, but you also do have actual primary data. So you have messenger RNA data from a specific data set, and there's a date from 2015. So this has been on there for quite a while. But you see that there's different messenger RNAs that have been placed here, and they come with the differences. Actually, you see maybe if you look carefully that there's slight differences here in the 3' ends. You see also differences here. So you see it's not that all the transcripts are ending at the same site, but you see very specific things. You see also differences in the 5' end here, and you actually see how some of these messenger RNAs coincide with some models, but other messenger RNAs coincide with other models. So you could probably imagine that these primary data would be kind of be the basis for someone deciding to make gene models which would have that structure here. I showed you already how you can show data on the genome, but you can not only show mRNA on the genome, as I showed before, but there's actually a lot of data which makes sense to see on the genome. There's genes. You want to have 5' data. So this track up here shows 5' data. So you see a peak here, which coincides with the start sites of this, with the start site of this gene, HCST. You would, for example, see a peak here, which coincides with the start site of that gene, and so on and so forth. If you basically highlight one of the peaks here in the Zenbu browser, you would see that you would get expression from certain samples here. You have certain cell lines, so these are data from the ENCODE project. But these are all from one specific cell line here. Down here you have a long RNA-seq, so you have RNA sequencing data, and the RNA sequencing data is also strand-specific, as is the 5' data, the cage data. So you see green are the reads that map to the plus strand, going into from left to right, and in magenta you see the reads that are on the minus strand. So for this gene here, you would have basically all of the expression on the minus strand. And if you look very carefully, you would see that the signals of the RNA-seq data, that they align well with the exons here. So you would have expression on the exons, of course. Down here you have a way, so this track here is a track which is composed out of one specific cell line. If you have data from many different cell lines, you can actually, or different cell types or cell lines, you can visualize them in such a heat map fashion. And if you do mouse over, which I intended here, you would see that each row corresponds then to one specific cell type. And you could see, for example, that for many different cell types, you have expression of that exon here and so on. So you can visualize RNA-seq data in various different ways, like in this way here or as a heat map. And you can also put expression into it. I try to make a slightly more comprehensive list of what you could actually show on the genome. And like in terms of models, we saw some genes and transcripts. You can also look at chromatin states. So chromatin states means that if the DNA is in a three-dimensional space, so it's like a one-dimensional structure in a three-dimensional space, and it is kind of wrapped in certain ways around histone proteins, which you probably know about. So you might want to indicate actually which areas of the genome are open, which ones are closed. So you can talk about the openness of a chromatin, which you can indicate. You can make contact maps. So you can say which parts of the genome are close to other parts of the genome. So this would indicate potential interactions between different areas of the chromosomes, or actually maybe even between chromosomes. You can look at topological domains. Usually the chromosomes are organized in that certain areas have frequent interactions, but these areas would not really have interactions with other areas. So you would have areas that are interacting a lot within, but not across these domains or areas. These are called topological domains, topologically associated domains. You can put models about gene regulations, about how transcription factors would bind, or like models of transcription factor binding sites, and about enhancers. You can display data itself. You saw some examples here of RNA sequencing data where you can focus on the 5' end or 3' end, or for the whole transcript. You can look at genome variation, so we can indicate specific positions where we have known single nucleotide polymorphisms, or SNPs, and they might be related to certain diseases. We can indicate DNA sequence properties, for example, which parts of that genome are conserved across species. We can indicate repeat elements in the genome. And here's primary epigenomic data. For example, we can actually indicate where real transcription factor binding, for example, has transcription factors are proteins that bind to the DNA, and it can be measured where these proteins are binding to the DNA by chromatin immunoprecipitation assays. So we can actually measure where proteins bound to the genome itself, or to histones which are at certain positions in the genome. This is slightly and also transcription factor binding, which corresponds to that. It's histones and transcription factors. These transcription factor binding models here, they are slightly different because they would result from scanning of motifs along the genome. Here the ATAC-seq data and DNase hypersensitivity data would be the primary data related to the openness of the chromatin. So we can show the data itself, but we can also show then how these data were used in a more comprehensive way to assess openness. So there are some derived insights that are shown in the models here, whereas these are the primary data, and the Hi-C and the capture Hi-C data would be related to contact maps or topological domains. Some words on the gene structure and the gene expression. So this slide is a short introduction which will lead us to the RNA sequencing, which I will talk about in a second. So it is good to understand the very basic principles of the gene transcription and splicing. So this is an attempt to visualize the DNA here in a one-dimensional way. And this is a gene which has its promoter, and then it has the green part, which is the actual part which would these are the exons of a messenger RNA, and there might be gene regulatory elements like enhancers inside. So the transcription, the first part of the transcription would lead to a premature messenger RNA, which would have the exons and introns in the premature messenger RNA. And then there is the step of splicing, where actually the spliceosome, which is a protein complex, which would actually remove the introns and would concatenate exons to each other. So that would lead to a mature messenger RNA with the untranslated regions maybe here and an open reading frame which for a protein coding gene then would translate into protein. So when we perform RNA sequencing, then it is actually that messenger RNA here that we are looking at. So it's the mature messenger RNA. So please keep in mind that what we are attempting is that we sequence these parts here, and we actually want to go back and we want to understand what kind of intron exon structure in the genome would these relate to. On the right side, we start here. You see basically something that I just explained in a more compressed way. That you have the premature messenger RNA and then the mature messenger RNA, which is spliced. So when we have then this mature messenger RNA, we would go ahead and we would fragment that messenger RNA after extracting it from the cells as a concept. And we would fragment these mRNAs into pieces in the very same or similar way as we would do for the genome sequencing before. There's a step of reverse transcription, so we would make a double-stranded cDNA out of these to make them more stable. And then we would have certain processes and we would sequence these parts here. And when you sequence these things, these RNA reads, then you have to align them to the genome, which is indicated here. And here I wanted to go back to a slide which I got from an NBIS workshop, which I find a bit more instructive, where you can actually map your reads. So these are the reads here, and you can map them to the genome. And if you map them to the genome, you have to be slightly aware of the fact that a read, which actually, if you map it to the transcriptome, it would map very close to each other because that region and that would be very close to each other on the transcriptome. But if you map it to the genome, you have to be aware of the introns. You have to have some intron-aware mapping here. So, and this way here, you would basically also learn about intron-exon structure. So you would see that certain exons would be skipped, which is indicated here for certain samples. It is very important to keep in mind that also when you sequence RNA, then you sequence RNA from a certain tissue or from a certain state. So certain messenger RNA variants might be present in certain tissues, but not so much in other tissues. Lastly, I wanted to show some efforts where consortia have made an effort to annotate the genome, and they try to put as much information as possible onto the genome sequence. Maybe the most well-known is the ENCODE consortium, Encyclopedia of DNA Elements. So they made annotations for human, mouse, worm, and fly. And it's about gene expression, about gene regulation, and they also make their own gene models, which are the GenCode models. And you see that their annotation is based on quite a lot of samples. It's like close to 20,000 biosamples, and you have different yeah, different samples here, and they have many different assays. So it's DNA binding assays, which is basically chip-seq assays, transcription, which would be RNA-seq, DNA accessibility, which would be a attack-seq or MNase treatment, RNA binding, DNA methylation, and chromatin structure, and so on and so forth. So I invite you to have a look at the ENCODE Consortium website. Another one out of several consortia is the Genotype Tissue Expression Project. This is in human only. And here the point is that this consortium look at the variation. So it's performing genome, maybe it's shown here, it's performing a genotyping or genome sequencing of individuals, human individuals, donors, and it's performing RNA sequencing at the same time. So you can make some expression analysis which is aware of the genotype, some eQTL, a quantitative trait loci analysis. So here they have data for around 54 individuals, 54 tissues, and around close to 1,000 donors, and quite a lot of samples. And here you see one example of how samples are expressed across tissues. So yes, I think with that I want to end this lecture here. Thank you for your attention, and we discuss further in the course.